Hi everybody, welcome back to class again. In this video, we're going to be finishing up the section on the scientific method and experiments. And again, if you haven't done so already, please go back and watch the videos to fill in your notes. And watching this video, please make sure you fill in your notes as you're going along. So in the last section, we were talking about conducting an experiment or testing the hypothesis. We're moving on to the step after that, which is collecting and analyzing your data. This is after you have ran your experiment, you have all this information. What do you do with it? Well, now you have to analyze it. But as you're collecting it before analyzing, please make sure that your data is specific, detailed, and accurate. Try to keep everything organized as well. Now, there's two different kinds of data you're going to be collecting, or you could be collecting. Qualitative data and quantitative data. Qualitative data is information based on the qualities of something. Qualitative data can be more opinion-based. For example, the table is smooth. Somebody may say it's smooth, somebody may say it's a little rougher. The dog is fluffy. The shirt's color is red. The star is really big. All of that is qualitative data. You're describing whatever it is. You could go along and say that the water is clear, colorless, transparent. You could say the glass is translucent. All of that is good data to know, but it is qualitative data. On the other side, quantitative data is information or data based on countable measurements of something. Length, mass, the amount of energy, the amount of force, how tall someone is. That would be your quantitative data. When you collect both of these types of data, it's very helpful. Think, for example, if you had to identify somebody who's walking around a park, having both qualitative data and quantitative data really helps out. If you know how tall they are, you can eliminate people. If you know what color shirt they're wearing, you can eliminate people. Now you can say, well, one person's red is different from another person's red. Yes, and that's one of the issues with qualitative data. But when you combine qualitative and quantitative data, it really helps out whenever you're analyzing. Now, when you are analyzing, make sure your data is neat and organized. Data that is all over the place, written in different places, written in different ways, is very difficult to understand because you might not even realize why you wrote it there. So make sure it's neat and organized. And you can organize it in different ways. As you see there, data tables, line graphs, scatter plots, bar graphs, or pie and circle graphs. The first two that I have here are data tables, and data tables and line graphs. And data tables are pretty straightforward. All you're doing is putting your data into a table. And it's the best thing about data tables is it's easy to read. In the example here with the plant growth and soils, you know which plant group you were working with, you knew the pH of the soil, and you knew the average plant growth. So it's very organized there. And you can just go straight down and you could draw conclusions based on that information just by looking at the data table. Line graphs are the same way. Line graphs are very easy to read. You can see trends. In the case of this graph, you know that as the week goes on, more people buy muffins, or there's more muffins sold as the week goes on. That's an easy trend to read. But if you had this data just written willy-nilly all over your paper, or it was in paragraph form, it's harder to read and harder to analyze. Now, with every graph, and I'm going to say this now, and please make sure that you record this in your notes somewhere. Every graph, or let's say line graphs, are going to have two axes. You have your x-axis and your y-axis. The horizontal axis is your x-axis, and your vertical axis is your y-axis. And your x-axis is your independent variable, and your y-axis is your dependent variable. So anytime you go to create a line graph, independent variable goes on the x-axis, dependent variable goes on the y-axis. And again, it says here line graphs are best for experiments that show continuous change over some period of time. Two other types of graphs are bar graphs and circular pie graphs. Now, these two types of graphs are really good when counting things. So with the muffins sold, you could have made that into a bar graph showing how many muffins were sold each day. That could have been made into a pie graph, absolutely. But with this graph at the line graph, they were trying to show a trend over time. They were trying to show you that as the week went on, sales went up, and it's easy to read. But you can also easily read a bar graph. Looking at the trees in the garden, right here, we know that there were more oak trees than any other trees because of the bar graph, easy to read. For favorite snacks, we know that fruit is the favorite snack based on the number of votes based on the bar graph. And you can make bar graphs that are vertical, meaning going up and down, or horizontal, going left and right. Either one is correct, it's just your preference. Now, circular pie graphs are also used for counting, but you usually see them show percentages. And that's just, you could just have them say here it has 3 hours, but it also has a percentage. They can have regular numbers, but a lot of times you see them with percentages. And both of these graphs are very good for counting things. After you have analyzed your data, you've collected your data, you've analyzed it, you put everything into graphs and tables, you can read them easily, you're ready to go through and draw a conclusion from all of this data. You're ready to make the decision. Was my hypothesis correct or was my hypothesis wrong? If your hypothesis was correct, you're going to test it again because maybe there was a fluke and you just got lucky. And you're going to see if you get the same results. And then you're going to test it again. Rule of thumb usually shows that testing it 3 times is like the minimum, but the more you can test it and the more it can be reproduced and the more you can get the same results, the better your hypothesis is going to be, and especially when you're talking to more people in your community, scientific community. And then after you have tested it multiple times, share your results with the community. Now, let's say your hypothesis was wrong. Did the results support your hypothesis? No. You have to decide how you could change your hypothesis. Okay, let's go with the tipping the water over. Okay, maybe it was a different type of glass we need to try. So now we're going to go back and try a different type of glass with it. Or you're going to try tipping it over a different way this time. Instead of just pushing it over, you're going to flip it completely upside down all at once. You got to find how are you going to adjust your hypothesis. A big thing, a big thing is find error. And there is always going to be some type of error. Human error is the most common. Find it and try to fix it and try to reduce it for the next time that you test it. And after you have changed your hypothesis and or fixed the error, test it again. If you're wrong again, go through, change something else, test it again. Again, when you look at the video back from a few days ago with the SpaceX rockets, how many times did they get it wrong and how many times did they have to keep changing something? And it can be absolutely positively frustrating when you're trying to prove your hypothesis correct. But the more you test it, the more you narrow down things that could go wrong and you keep fixing them until eventually you get it correct. So the last part of this notes here is another example experiment. In this experiment, it says Bob wanted to investigate the effects of fertilizer on plant growth. He bought 2 identical plants. He gave plant A 50 milliliters of water every day and plant B 50 milliliters of water every day and fertilizer every day. He set both plants in the same spot on the porch to get the same amount of sunlight. He measured the height of the plants once a day at the same time and recorded his results in the data table. It should say to the, not to the right, to the left over here. That would be his data table. So what would have been his hypothesis? Based on this information, what was his hypothesis? A guess could be, whoop, that doesn't work very well. Let's go to purple. If plants are given fertilizer, then they will grow taller or larger. And that's a testable hypothesis. So based on this hypothesis, and could you have a slightly different hypothesis? Absolutely. But make sure it matches your experiment. He wanted to test the plant growth. Make sure your hypothesis matches both fertilizer, effects of fertilizer, and plant growth here. I forgot that I could highlight on this. Your independent variable then would be with or without fertilizer. So giving or not giving fertilizer, or you could just say fertilizer. Your dependent variable then would be plant growth, or I set up here specifically plant height. So, based on this information, what would be your control? Your control would be the plant width, and I apologize if I haven't told you this already. When you see me write W with a slash, that means width. It's shorthand for me. And when you see W/O, that means without. So plant without fertilizer. That would be your control. You're comparing the effects to fertilizer to no fertilizer. Constants in this experiment were the amount of water. The constants were the plant type, the location that both plants were, the time that they were measured. So these are all different constants. And you could go through and say there's more constants. You could say that there could probably be the soil. You could say the, let's say, type of water. So he doesn't give one plant spring water, another one tap water. All of those could be constants. And you could probably, as you keep going through this, have even more nitty-gritty constants in here. And then based on this information, what would be your conclusion? So to have our conclusion drawn, we got to go up to the data table. And we have to see. On day 1, the plants were the same height. On day 2, the one with the fertilizer grew slightly more. It had 0.1 centimeter more growth than without fertilizer, than with only water. And as we go down the chart there, the easy to read data table, we can find that every day the plant with fertilizer grew taller than the plant without fertilizer. So the conclusion is fertilizer will make plants grow taller. Or the type of fertilizer would make them grow taller. And we forgot that in constants where it could be the type of fertilizer. So whenever you go to draw your conclusions, make sure you look at all your data. Make sure you're looking at how your dependent variable was affected by your independent variable. That's all the notes for this section, everybody. You, again, will be getting practice with this material. This means now that we finish, you will have a test coming up. You will have a quiz coming up. So please make sure you study. Please make sure you ask me any questions that you might have. Thank you, everybody, and I hope you